Everyone. I'm Dr. Neil Kelman, the chair of family medicine at Mount Sinai. And if you're anything like me, people all day long are asking you for your opinion about what's going on with COVID and breakthrough infections and whatever. And I feel like the best thing that we could possibly do was have our international expert, Dr. Florian Krammer, who is the chair of the Department of Virology at Mount Sinai, rejoin us again. You may recall, for those of you who are lucky enough to hear him the first time, how much we learned about the COVID virus, and we are thrilled to have him back. Dr. Krammer graduated from the University of Natural Resources and Life Sciences in Vienna, Austria. He received his postdoctorate training in the lab of Dr. Peter Police at the Icahn School of Medicine at Mount Sinai. In 2014, he became an independent principal investigator and is currently the Mount Sinai Professor of Vaccinology at the Icahn School of Medicine. Dr. Krammer's work focuses on understanding the mechanisms and interactions between antibodies and the viral surface glycoproteins, and on translating this work into novel, broadly protective vaccines and therapeutics. His main target is influenza virus, but he's also working on coronavirus, flaviviruses, hantaviruses, filoviruses, and arenaviruses. Since 2019, Dr. Krammer served as principal investigator of the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, which is trying to develop a long-lasting permanent influenza vaccine. And if I remember correctly, I believe the dean at one point said this grant was the largest grant ever received at Mount Sinai for a single research endeavor. And so it's really a thrill to have you back again, and we can't wait to get updated on everything you have to tell us. Dr. Krammer.
Thank you very much for this very nice introduction. I'm going to try to share my slides here. I think it should work. Before I start, I just wanted to say it would be great if you could hold on with questions until after the presentation. I have a lot of slides and I want to make sure we get through them. And one slide correction, Peter Police is still my chairman.
Oh, sorry.
But no problem.
And just so, put your questions in the chat if you would, and then we'll try to get to them at the end of the presentation.
Great. Okay, so just very briefly, my conflicts of interest here. Of course, we have been very active in the area, and together with PwC and Adolescents here, Austria, we have developed COVID vaccines. I'm not going to talk about them today, but they have been developed in middle and low-income countries. They're currently in phase 3 trials in Vietnam and in phase 2 trials in Thailand, Mexico, and also in Brazil. And so I'm named as inventor on these IP filings. Just as a very brief refresher, which types of COVID vaccines do we have? And you might come across some that are actually not licensed in the US. If you look worldwide, we have inactivated virus vaccines that are used. So this is just SARS coronavirus 2 grown and inactivated, like hepatitis A vaccine, for example. We have the mRNA vaccines, which you're by now very familiar with. That's what is mostly used in the US. We also have viral vector vaccines. You're also familiar with that. In the US, we use the J&J vaccine that's based on viral vectors. And then we have recombinant protein vaccines. Some of those are now licensed in certain countries. And we're still waiting in the US for seeing Novavax recombinant protein vaccine to enter the market. Sorry. So these are the main types of vaccines that we have. And just as a refresher, what did we learn about these vaccines from the clinical trials? So the Moderna and Pfizer vaccines, which are both mRNA vaccines, had very high efficacy in the clinical trials. The Pfizer vaccine is now fully licensed in the US. The Moderna vaccine is still under an emergency use authorization, but should be fully licensed soon as well. And then we have the J&J vaccine. As I said, that's a viral vector vaccine. And the special thing here was that it was only given once, while the Moderna and Pfizer vaccines had to be given twice. And then in Europe, we also, and globally, we also have the AstraZeneca vaccine, which is getting used a lot. It's actually the vaccine that is used the most globally. Novavax, as I mentioned, in the US is still in development. In other countries, it has been licensed. And then we have viral vector vaccines from Russia and from China that are used globally, and inactivated vaccines from Chinese companies as well as from Indian companies that are used globally too. And so I'm giving you here the efficacy numbers for these vaccines from the clinical trials, but we'll discuss a little bit later what that means now for Delta and how well these vaccines still work in the population. Before I go into vaccines, I just wanted to give a brief update about protection after natural infection. And this has become, at least in the US, a pretty politicized issue. And I wanted to show some data to provide clarity here. I'm going to show you some data from our studies at Mount Sinai, and then I'll point out some publications that you could refer to if you have, that you could look to if you have questions. So some of you might know we have a large cohort study ongoing at Mount Sinai that's called PARIS, or Protection Associated with Rapid Immunity to SARS-CoV-2. We started this cohort in April 2020, and we enrolled both naive individuals into this cohort and people who already had suffered from SARS-CoV-2 infection. Remember, back then we had a lot of infections. So it's about half-half in terms of people in the study. And these are mostly healthcare workers who work at the main campus at Mount Sinai. And we follow those people for antibody kinetics to see, how the antibody levels or the immunity develops over time. We bring them in at least once a month to get bled. And of course, if somebody is getting an infection, we also bring them in. We basically document that, and people are also tested occasionally for viral RNA in some of the samples that we take, that it includes saliva samples. And this allows us to look at the reinfection risk and the protection that people get from natural infection. And so we recently did an analysis of the data that we had before people started to get vaccinated. And on the right side, this is in a way graphically shown. So in this part, we had 246 seronegative individuals, which are these blue spots here. And we had 154 seropositives, which are the orange ones. And we detected 11 infections in that cohort during that time. 10 of them happened in naive individuals. You see here this teal color. This is basically an infection. And we had one infection in a person who was previously infected with SARS coronavirus 2. But at the time of when this infection occurred, this person had lost the antibody titer. So this was a serorevertant. And so from that study, we can conclude that having been infected and having antibodies, having a detectable immune response, does protect you from infection. And this is not just something that we observed. There's a lot of studies out there that show the same, basically showing that natural infection gives you about the same level of protection as vaccines do. As I said, there are a number of studies out there for the pre-Delta era, but there's now also a study out from the UK that was done in the post-Delta era, and that basically shows the same, about a 75 to 80% protection from infection with Delta. And that brings me to an important topic, and that is, how does our immune system protect us against SARS-CoV-2? And in the last few months, we have learned a lot about that, specifically in terms of quantity. And I wanted to explain that a little bit. First of all, there are differences between what an infection does in terms of inducing immunity and what the vaccine does. So when you get an infection, you're exposed to all kinds of proteins of the virus, the structural proteins, which you see here in this schematic of the virion, but also to the other proteins that the virus makes in our cells. There are many additional proteins that are expressed. And so you get an immune response against the spike, of course, which is important because these antibodies against the spike can neutralize the virus. You get a lot of other antibodies against other proteins, and you get T cell responses across the genome or across the open reading frames. And what is also important to say here is that when you get infected with a virus, viruses are also always quasi-species, so they're not monoclonal. So, you know, one virion might have a mutation in the spike, another virion might have a mutation in another area of the spike, and so the immune system is exposed to a little bit more variety. And of course, because this is a mucosal infection, we get systemic immunity, but we also get mucosal immunity. For the vaccines, on the other side, we basically only look at the spike, except for inactivated vaccines. There's one consensus spike sequence, so there's no diversity like in infection, and you get systemic immunity, but no mucosal immunity. Now, there's also a quantitative difference. Typically, when we get infected, the immune response is not as strong, and it's also more heterogeneous. Some people might actually respond very well, and other people might respond very weakly. For the vaccine, we know that healthy adults always respond very strongly. So there's pros and cons. I'm not saying one is better than the other, but there's differences that we need to understand. So what really protects us from infection? I want to get to the point of correlates of protection because I think this is important and underappreciated at this point. So there were very early experiments. This is from Dan Baruch's lab, where they basically took serum from non-human primates that were infected with SARS-CoV-2. The seroconverted made neutralizing antibody, and then they took serum from these animals and put that into new naive animals in a dose-dependent manner. You see here 250 mg per kg in terms of IgG, 25 mg per kg, 2.5, and then 0. And then they challenged these different groups of animals with SARS-CoV-2. And if you give enough antibody, the animals are completely protected. If you give less antibody, protection goes down a little bit. And at some point, it doesn't help anymore because there's too little. So we know that antibody alone can actually protect these animals. And that's probably through neutralization. But there's also other antibody functions. Antibodies can actually activate macrophages and natural killer cells and lead them to kill infected cells. So this is in non-human primates, but Moderna, when they were Regeneron, when they were developing monoclonal antibodies as prophylactic treatment for SARS-CoV-2, they did the same experiment in humans. And so they had a clinical study where they enrolled families. And when one person in the family tested positive for SARS-CoV-2, they gave monoclonal antibody, neutralizing monoclonal antibody to the other family members. And then they looked in how many cases do we see transmission to the other family members. And then they looked at ratios when that happened in the placebo group versus the antibody treatment group. And so they see very high efficacy of these antibodies given prophylactically. And that also hints towards antibodies being able to prevent infection just on their own. We have additional data now. There were studies that looked at the vaccine efficacy in clinical trials of different vaccines, and they compared that vaccine efficacy or tried to correlate the vaccine efficacy to the antibody titers that were achieved with these different vaccines. And there were two nice studies that came out. Here on the left, you have one that was published in Nature Medicine. And here on the y-axis, we have the vaccine efficacy. On the x-axis, we have the neutralizing antibody titer, and this is normalized here so that you can compare it. And you see that there's a nice correlation. The higher the neutralizing antibody titer, the better the efficacy. And the second paper basically showed the same, even also with binding antibody here on the right side. So it doesn't even have to be neutralizing antibody. And very recently, this is now out, there are two preprints out. People have looked at the actual titer that you need to achieve in order to get a certain risk reduction. And this is shown here nicely on the right side. Here we have on the x-axis antibody titer. This was a specific neutralization assay, but this is not not really important here right now. And on the y-axis, we have the probability of getting infected with SARS-CoV-2. And so here you have basically no antibody, and this is your placebo, the placebo control group's probability of getting infected here. And then here, you see this curve basically going down here. So the probability of acquiring an infection is sinking the higher the antibody titer goes. And so this is, I think, very important, because that now indicates that higher antibody titer in general means that the probability that you get infected with SARS-CoV-2 is lower. And I think that's an important step forward. And I hope that we will soon have units, international units that will be specified, that they can tell you if somebody is still well protected or if somebody is not protected enough anymore. Of course, I don't want to only focus on antibodies here. The same paper that showed that antibody alone can do it also showed that if the antibody levels are very low, T cells become more important. This is shown here on the right side. In animals that have lower antibody titers, if T cells are depleted, then you start to get breakthrough infections. So T cells also have a very important function, but antibody alone can do it. And just to make sure that everybody has seen that data, vaccination also induces relatively strong T cell responses, and those are mostly CD4 T cell responses. The CD8 T cell responses are typically a little bit lower, which is consistent between infection with SARS-CoV-2 and vaccination against SARS-CoV-2. The problem that we are facing, and this is actually a very nice study from Mount Sinai, which was conducted by Samir Barek with Viviana Simon and Anja Wannberg. The problem that we're facing sometimes is that people are immunosuppressed and cannot make a good immune response after vaccination. There's data for one patient here who had multiple myeloma, got vaccinated twice, and then got infected and unfortunately died. And it turned out this person didn't make any antibodies or T cells, and that's often correlated. In general, we find that multiple myeloma patients do not, 15% of them do not respond to the first two vaccinations, and then their antibody titers go down relatively quickly. But the third dose, if you give them a booster, that usually helps. But this is something to keep in mind. If somebody doesn't make an antibody response, there is really an issue, and that needs to be addressed. If you want to test for antibodies, typically there are two types of tests, against the spike protein and against the nuclear capsid or nuclear protein. If you want to look for success of vaccination, meaning if there is an immune response, the nuclear protein based tests are not helpful because the nuclear capsid or nuclear protein is not in the vaccine. So you need to use a spike based assay, and ideally that assay should be quantitative and should have a readout in international units or BAUs. The FDA still says that antibody tests are not useful. The CDC changed their mind, and they actually have a lot of recommendations online about how to use antibody tests to the benefit of patients. And this brings me to the more recent updates. As you all know, in spring of this year, it started to look really good. The vaccination rates went up, the case numbers went down. This is shown here for the US in general, but the same happened in New York City. And then we were all thinking, oh, it's going to be over soon and we're going to be good. But then in June, Delta started to circulate in the US, and this is shown here on the right side. This is the proportion of Delta infections for all SARS-CoV-2 infections, and you see how Delta kind of explodes here, and that led to a wave in the summer, and now it was going down a little bit, but now we see a little bit of an uptick again. And so why did that happen, and what do we know about Delta? The first question, of course, was, now we have this Delta variant, do the vaccines still work against Delta? And the first thing that was done was, of course, looking at neutralizing antibodies of, to see if sera from vaccinated individuals still neutralize Delta. And this is a study that Viviana Simon and I did here at Mount Sinai. We looked at many different variants. So here is neutralization against the wild type. We set that to 1. And then when we look at Delta, which is here, we see a 3-fold induction in neutralizing activity. So that's not too bad, actually. That's not too much of an escape phenotype. The worst is usually the Beta variant here. And we also had a Lambda subvariant that was doing bad. But basically, yeah, there's a reduction in neutralizing activity, but the neutralizing activity is certainly not gone. So from that perspective, we should still be good. And I'm going to review a little bit the data that we have in terms of how effective the vaccines still are in the population. But before I do that, I wanted to explain efficacy versus effectiveness, because if you listen to the media, they're using both interchangeably, and there's a lot of confusion about that. So efficacy is what we get from the clinical trials. How well does the vaccine protect in a controlled clinical trial setting? And this is, of course, done in a defined and healthy study population. For COVID, we also had older individuals that were included, but in general, a healthy population. They're strictly controlled vaccine administration. These groups are highly monitored, and we have a placebo control group. And this gives us efficacy. Effectiveness is something very different. That's basically measuring how well does the vaccine work in the population. And this is basically an undefined study population with people with all kinds of comorbidities. The vaccine administration is not controlled necessarily. The timelines are not necessarily always adhered to, and there's certainly no placebo control group. So, you know, you need to become, you need to use different study designs in order to calculate how well that works. It's usually a test negative design that is used there. So these are two very different things. It's apples and oranges. And then there's another important point, and this is endpoints. So for the original efficacy trials, Pfizer and Moderna's endpoints in the trial were any disease plus PCR positive confirmation that somebody had really SARS-CoV-2. And for J&J, this was different. It was moderate to severe disease. So that's already a very different endpoint, right? And then if you look at the effectiveness studies that are conducted now, many places, I mean, there's many effectiveness studies that have been published, they usually look also at protection from infection. And this is not something that was evaluated in the clinical trials. So we have to be careful if we hear numbers, percentages, that we understand what that means, because we might be comparing apples and oranges. Okay, so there's one study out there that has data, and that's the Pfizer study about efficacy against Delta. And this is what I'm showing you here. So they looked at basically efficacy right after the second dose, and then they followed that over time, and you have a little bit of a drop from 96 to 90%. And then with the more than 4 months, they basically also entered the Delta era, and you see that there is a decrease in efficacy down to 83.7%. So that's not too much data, and we're waiting for more long-term data from that study. But in the meantime, there were, I think it's more than 40 effectiveness studies now that really looked at Delta. And I'm just summarizing this here, and then I'll show you one specific example. So it looks like the effectiveness against infection, which was somewhere around 90% in the beginning when Delta was not circulating yet and when people were freshly vaccinated, went down to 50 to 80%. The effectiveness against symptomatic infection is mostly still between 80 to 90%. Some estimates are as low as 65%. So it varies. It depends a little bit on the study, but there was certainly a drop, and you will see that as well in my next slide. But the most important thing is the effectiveness against hospitalization stayed high. That didn't drop. And I think this is nicely shown here. This is a study from New York State, where they actually looked at vaccine effectiveness every week from May until July. July is already, this was all Delta. But in the beginning, there was no Delta around. And so they looked at two measures: effectiveness against infection. So this includes asymptomatic infections and symptomatic infections. And they start with 91.7%. And then you see this starts dropping, and it drops down to about 80%. It almost looks like it plateaus here. And then on the other side, we have effectiveness against hospitalization, which was a measure for severe infections. And they start here with 95.3%, and they end with 95.3%. And I think this is really the most important point here. People are still very well protected from severe disease, even though the vaccine effectiveness against infection went down. And when I say went down, I don't mean it went away. There was just a reduction. 80% is still a good number. So why is this happening? There are a couple of factors. There are viral factors and there are human factors. The viral factors are Delta is more infectious, and I'll actually show a little bit about that. Delta replicates better in the upper respiratory tract. We have usually much lower CT values and higher viral titers as compared to the other variants. Delta may be faster in getting into cells. The spike protein gets better activated, and it might enter cells quicker, and that helps it to escape the immune response a little bit. We know that there's a partial immune escape phenotype. It's not strong, but it's there. And Delta has a shorter incubation time, and that's also important because in many cases, when you have a long incubation time, an anamnestic response might actually prevent disease. But if you have a shorter incubation time, the immune system doesn't have a lot of time to respond, and that might also be a problem. And then, of course, we have human factors. First of all, there's some waning of the immune response. I'll show you data about that too. There's behavioral changes. People don't, I think in New York, we're still pretty good, but there are other areas where nobody cares about the countermeasures anymore. Nobody cares about the virus anymore, and life goes on as normal. And that, of course, also helps the virus. And then there's, of course, also, I told you about this test negative design. There's also a mathematical reason why vaccine effectiveness goes down. And that is because a lot of the people in the negative control group basically had a natural infection. They might not be vaccinated, but the natural infection also has a protective effect. And so mathematically, there might also be a little bit of an impact. I said Delta is much more infectious, and I want to show this here. This is based on data from the Lancet. The original virus from Wuhan had an R0, or a reproductive number of somewhere between 2 and 3. So one person infected about 2 to 3 people. This here is Delta. In Delta, one unvaccinated person infects about 5 to 8 unvaccinated people. This is not as bad as measles, for example, which is really, really infectious, but it's already bad, right? It's much worse than the original virus. And that is, of course, a problem. And just to show that graphically, if you have this one unvaccinated person getting infected, that person will infect potentially 8 other people, and 7 of them will most likely become symptomatically sick. And these people will transmit each to 8 more people, right? And so this ends up to be a lot of infections. However, if you're vaccinated, you can still get the disease, but at a much lower rate. So here you have a 70% reduction. This is an unvaccinated person. These are vaccinated people, but only a proportion of the vaccinated people get infected. And then even the spread to unvaccinated people would be reduced from these 3 infected people here. So there's a huge difference what the virus can do in a vaccinated population versus what it can do in an unvaccinated population. It's certainly true that people can have breakthrough infections and they can also transmit, but the rates are very, very different. And then I also wanted to show you some data about immunity, and this is again from our studies at Sinai. What you see here are basically antibody titers over time. So you on the x-axis you have the time, on the y-axis you have the antibody titers. And the orange dots are people that we enrolled that had already had an infection and then were vaccinated. And in blue, you have people who were naive and then were vaccinated. And so I think you can see it best on the right side. Of course, the people who already had an infection start off with a higher level. They get their first vaccination, the titers shoot up. The second vaccination doesn't help them that much. And then they start to have a drop of about 10 to 20 fold in antibody titers. And then there's a phase where they start to stabilize. And the same is true for naive individuals. The dynamics in the beginning are different. The first shot gives them some immunity and the second shot actually boosts that quite a bit. But then you see the same drop. And then you see also a stabilization phase. I'm showing a better graph of that in a second. And the people who had the previous infection were vaccinated stay, of course, at the higher titer in general, but the dynamics are kind of similar. The decay rates are very similar. And what we look at here is the fold decrease from basically 1 month to another. And you can see that initially there's a steep decline in antibody titers. Again, here we have about 16-fold, but there is a stabilization phase. So, and that makes sense from an immunological perspective. So even people who got the vaccine twice at some point will have stable titers. And we hope that that will correlate with stable protection against infection. Maybe not at 95%, maybe more around 80% or 70% as we have seen, but that would still be robust. Another point that I wanted to make here is there's often the question, oh, I got already an infection, should I even get vaccinated? And the clear answer to that is yes. The reason for that is, as I mentioned earlier, the immune response after infection is pretty heterogeneous. You see that here. There's some people who even lost their antibody titers. There's some people who have really high ones, and there's a lot of variability in between. But what happens if these people get vaccinated? They get really high and homogeneous titers. And they basically get, compared to just vaccinated people, a super immunity, right? And so that's one of the reasons why it's highly recommended that people who had a natural infection still get vaccinated. We think one vaccination is probably enough. In the US, the recommendation is still to vaccinate them twice. But in Europe, they changed that, and people who had a natural infection only get vaccinated once. And that's the nice thing is that's partially on data that we provided in the beginning of this year. The other benefit here is, so we of course also monitor breakthrough infections here, and in the vaccinated individuals we have found 14 breakthrough infections after in fully vaccinated individuals, but we haven't found a single breakthrough infection in those people who had a natural infection and then got vaccinated. So there are a lot of reasons to get vaccinated, even if you had a natural infection. I'm showing this because this might be interesting for you. We also look at differences in antibody titers between Pfizer and Moderna, and the Moderna titers are in our hands on average about 35% higher. So not really much of a difference, but it's probably because Moderna has a higher dose in their vaccine. Okay, so I'm going to switch the topic a little bit, and I'm going to talk about vaccination during pregnancy and vaccination of children. For pregnancy, I'm not going to say much. The data is solid. There are several large studies that evaluated safety in pregnant women, and the vaccines were found to be safe. One of these studies is actually a CDC study. The CDC explicitly recommends vaccination for pregnant individuals in all trimesters, and the same is true for the American College of Obstetricians and Gynecologists and the Society for Maternal and Fetal Medicine. There's a recommendation for that. There's no doubt about the robustness of the data, and pregnant women should get vaccinated. But there's a lot more to say about vaccination of children, specifically because there's a lot of data now and there's a lot of excitement about it. And I wanted to ask with the question, do we even need COVID-19 vaccines for kids? And so I wanted to go through the data a little bit. And you might actually have more experience with that than I do, but I tried to review what is out there and what we know. So typically what we see, and this is true for case fatality rates and infection fatality rates, which are both measures of, what percentage of the population dies when they, or of patients die when they get infected. We see that, of course, the risk is much higher in older individuals. The graph on the left is case fatality rates from a very early study in China with basically a lot of participants. And we see that, the lower the age, the better people are dealing with the SARS-CoV-2 infection, and then the case fatality rate goes up if people get older, and we don't even have under 10-year-olds here. On the right side is a graph from somebody who collects data on different infection fatality rate studies that are stratified by age and compares that to what we see for seasonal influenza, which is here shown in blue, and that's CDC data. And we see the same thing. The older you are, the worse it is in terms of infection fatality rate. The infection fatality rates, even in kids, are higher than for influenza, it seems, but still, these are low infection fatality rates. But still, what we have seen in the last few months, specifically with Delta, was that the number of hospital admissions of under 18-year-olds went up. This is kind of, if you look here at the data, this is actually from a summary from the New York Times. This is the Delta wave, and these are hospital admissions of under 18-year-olds. And this seems to be specific for the Delta wave, and that might be just because the virus is more infectious. Of course, if you compare that to hospitalization rates in other age groups, it's still little, but it has increased. And I think there was a story in August that said that about 30,000 kids had been hospitalized in August during the Delta wave. And the other important point here is that Delta is much more infectious, as I already said, and it's also really driving in infections in kids, and kids seem to be much more involved now with Delta in spreading the virus in the community. This is just basically data, again, from the CDC of basically outbreaks in different counties. This is before school started, and this is after school started. They looked here at schools using masks versus schools not using masks. That's not the point here. The point is school started and the infections went up. And so we have to keep that in mind too, that the children are now more than before a driver of community spread. And so for me, there are two legitimate reasons to vaccinate children. One is to protect them from infection and disease, including MIS-C. And then the other point is to protect their communities from onward transmission. So what do we now know about COVID-19 vaccines in children? Are they safe and efficacious? And so I'm going to start with what we know about the vaccines in older individuals, in adolescents. The vaccines, as you know, the Pfizer vaccine is now used in that age group. There were phase 3 studies that have been conducted to look at that. The first one here is with the Pfizer vaccine, where they had about 1,100 placebo recipients and 1,100 vaccinated individuals. And they looked, of course, at side effects, which were similar as compared to a group that was slightly older. And they also looked at immune responses, and the immune responses were similar to an older group. And they calculated a vaccine efficacy of 100%, which of course you have to take with a grain of salt because there were very few infections. But we can assume that the vaccine efficacy is similar to what we see in adults. Moderna did exactly the same and found exactly the same, had exactly the same results. Basically, a vaccine behaved very similar in the age group 12 to 18 in this case, or 12 to 17, as compared to older age groups. And they also found a very high vaccine efficacy of 93.3%, which again is very similar to what they found in adults. And of course, there's now a lot of experience with these vaccines because they have been used for months now in that age group. And so basically, When we talk about the Pfizer vaccine, we're talking about the age group of 12 to 15 because the Pfizer vaccine was from the get-go routinely used in 16-year-olds and older. And for Moderna, we are talking about 12 to 17 because their initial emergency use authorization was 18 plus. In these individuals, the regular dose is used, and by now about 50% of the, I think about 25 million 12 to 17-year-olds in the US have been vaccinated. Of course, there was safety monitoring. There are always things that you don't find in a phase 3 trial because they're rare and then you cannot detect them in a few thousand kids. But what was found was that we have about 40 cases of myocarditis per 1 million vaccinated 12 to 29-year-old males. And this is interesting because it seems to be sex specific, so it doesn't really happen in females. And so the Pfizer-BioNTech vaccine is currently used in the US in the age group 12 to 15 in the US and Europe. And actually in Europe, Moderna is currently also used in that age group. However, some countries, specifically Scandinavian countries, are only using it in older adults now because they are worried about the Moderna safety in terms of myocarditis in the younger ones. So they only use Pfizer in that younger age group. What do we know about vaccination of younger children? Vaccine trials for kids from 6 months to 11 years of age are ongoing. Pfizer initially tested 10, 20, and 30 microgram doses. Just as a reminder, the adult dose is 30 micrograms. And then they refocused their studies and tested 10 micrograms in the 5 to 11-year-olds and 3 micrograms in the 6 months to 5-year-olds. We'll talk about the Pfizer results in a minute. They released them in September, and this is now approved and ready to be used. For the younger age groups, we are expecting results in November. We'll see. They said maybe end of October. We didn't see any results, but they are now saying November. And Moderna also tested a reduced dose, so half of the adult dose, and has now results for the age group 5 to 11 as well. What do we know about the Pfizer study? This was a phase 2/3 study in approximately 2,300 participants. 10 microgram dose was given twice, and then they compared that to data from 16 to 25-year-olds who got the full dose, and they produced the same antibody titers, and it was also well tolerated. So it was similar to the full dose in a little bit older kids. And of course, they submitted the data package to the FDA, and now FDA and also ACIP concluded that the benefits outweighed the risks. I didn't put all the data here, but if you're interested, the FDA and Pfizer data can be found on the FDA homepage. The VRBPAC meeting where they discussed it is on YouTube if you want to watch that. It's 8 hours, by the way. And then the data was recently also published in New England Journal of Medicine. It's not just in the US that we are starting to use vaccines in kids. I just wanted to give an overview of what's happening worldwide. CoronaVac, which is an inactivated vaccine, is approved for use in 6-year-olds and older in Chile, 3-year-olds and older in China. There are studies with CoronaVac in children ongoing in South Africa. Sinopharm, which is another inactivated vaccine, is used in children 3 years and older in the United Arab Emirates. Cuba is using the Soberana 02 vaccine from the age of 2. The AstraZeneca trial in children was halted in April, and the status is kind of unclear right now, so they don't seem to go forward. And J&J seems to plan a study in 12 to 17-year-olds, but I'm not sure how well that is going and if they already initiated that. So to summarize vaccination in children, COVID-19 vaccines in children and adolescents are useful and seem to have a positive risk-benefit ratio. At least that's what the FDA and ACIP concluded. The Pfizer-BioNTech and Moderna vaccines are used already in adolescents and appear to be safe and effective. The Pfizer vaccine can now be used for the age group 5 to 11 at a reduced dose. And I want to point it out, this is a different formulation. It's not the same formulation as for adults. It's a different dilution. And it also has different stability data, so it can be stored up to, I think, 10 weeks in a fridge. So it's not the same vaccine formulation. Studies in younger children are ongoing, and inactivated vaccines are also used in children outside of Europe and outside of the US. And that brings me to my last point here, which I will cover quickly so that we still have time for questions. And this is booster vaccinations. And a lot of people have questions about that. And so I wanted to cover it and also show you some data that we have. This is actually a graph of one person. This just really nicely shows what happens after booster vaccination. So this is one of our trial participants. And basically what you see here is the person got vaccinated initially twice here, and you see the titer shoots up. Then there is waning, the titer goes down a little bit, the titer stabilizes over time, and then there's a booster dose. And so the titer goes up again a little bit higher than what we saw here. And then the big question mark is now what's happening now? What is happening now? Is the titer going down again to this same set point, or is it going to stay higher? And that's really the most important question about the booster doses. Just a few general things. And this is my opinion. That's not necessarily the official guidelines. I think booster vaccination is important for older individuals, individuals with compromised immune systems, and individuals who got the J&J vaccine. And for those individuals, it's important for maximizing protection. I think it's also important for healthcare workers to maximize risk. Sorry, there's a typo. To maximize risk reduction of virus transmission to patients, right? You've seen that the protection against infection goes down, and that means that you might be very well protected from disease, but you might still get infected, and then you might onward transmit the virus to a patient. And I think the booster dose really helps well to minimize that risk. For everybody else, I think boosters are a good idea if there is a high incidence situation like European countries right now, but might not necessarily be needed for everybody if there's a low incidence situation. And the 6 month interval that is used right now makes a lot of sense for getting an optimal booster response, except for J&J vaccine recipients, because I think they should get the booster much quicker, and for people with a compromised immune system, where a booster might be given relatively shortly after the second dose as well. Just to show you some data here, again, these are the same curves as before, but now we separated out the people who got the booster dose here in green. So these are all naive individuals who got 2 shots and then in the end got a booster dose, and you see how this shoots up again. It's actually kind of ending up at approximately the same level here. This is a more detailed look on that. Here we have basically what happens directly after the booster. People have all kinds of titers pre-boost, and then they get the booster dose and they all shoot up. From these very few individuals here, and the data is, you have to take that with a grain of salt, it seems like the titers are more stable than after the second shot. And that would be great if the titers really stay up like that. That would probably mean that there would be long-lasting, very high protection that wouldn't go down. But because we only have a few individuals that go out that far, I'm a little bit afraid to say that we will have long-lasting high immunity after the booster. We'll see how this develops. But so far, I have hope that that might actually be the case. The other interesting thing is I showed you earlier that people who had a natural infection and then got vaccinated typically have higher titers as compared to people who just got vaccinated. But then when you boost these people, they now finally end up at the same titers, right? So as you see, the people who got a natural infection and then get vaccinated have higher titers as compared to individuals who were naive and got vaccinated at the pre-boost time point, and then when they get the booster, they go up, and you see that they end up at the same peak titer. That's the first time that we see that. And this is just shown here again, graphed differently. So this is basically absolute titers here. In orange, we have people who had a natural infection, then got vaccinated. and this is here the pre-boost time point, and this is the post-boost time point. And in blue, we had people who were naive, got vaccinated, and now are getting the booster dose. And you can see the people who were only vaccinated profit in a way more from the booster dose because their increase in titer is higher, while people who had the natural infection and then the vaccination, then when they get the booster dose, they don't shoot up that much anymore because they're already high. And so it's kind of, the third dose is kind of an equalizer here. This is shown here again. And here on the right side is just the fold induction over the pre-boost level. What is maybe interesting for you, and you might come across these cases, is how does this now compare to a breakthrough infection? So if somebody got vaccinated and then got a breakthrough infection, does this person still need a booster dose, or is that natural infection inducing big and strong enough immune response? And so on the left, I'm showing you the same data that I just showed you. It's just more colorful. And these are people who got vaccinated. It's a mix of infected, vaccinated, and just vaccinated before the booster dose and after the booster dose. And here we have 4 individuals who got vaccinated and then got a breakthrough infection. And you can see that the titers go equally up after the breakthrough infection. So it's very likely that these people don't need a booster dose because the natural infection that they, or the breakthrough infection that they got, acted already as booster dose. And so with that, I summarize what I presented here today. Vaccines against SARS-CoV-2 work and protect well against disease. And this is still the case with Delta. Data from 10,000s of people in phase 3 trials, and now we can say billions actually, billions of vaccinated people suggests the vaccines are very safe, including in adolescents and children. And individuals with previous infection also benefit from vaccination, although only one shot may be needed. And the last point is, and we see that more and more, titers matter. It matters how much immunity you have. And I think in the coming months, we will actually see guidelines how to deal with that and how to measure that accurately. And with that, I'll stop. I just wanted to thank a lot of people at Mount Sinai who contributed to collecting the data that I just showed you. And specifically Viviana Simon, who is my partner in crime here for the Berry study and several other studies that we're doing. And with that, I'll stop presenting, and I'm happy to answer any questions that you might have.
Yeah, while we take a look at some of the questions, I just wanted to ask you about the news that's come out about treatments and whether or not that's going to change. You expect that that will change anything that we do around immunizations, especially for people who might be immunosuppressed. Could you imagine a time where we're giving people like prophylactic medications? And what about the antiviral medications?
Yeah, that's a very important point because we still don't know what to do with people who don't respond to a vaccination, right? And that's really important. There are a number of options out there now. First of all, of course, if somebody who is high risk and didn't respond well to vaccine comes in and with a confirmed infection, and it's still early on, we can treat that person with monoclonal antibodies, and that works well. It needs to be done early. The monoclonal antibodies don't work well if they are used late. So if the person is already severely sick, the antibodies might not have any effect anymore. But if you do that early, you might actually basically, this might result in very mild disease that is very transient. And we're getting additional options other than monoclonal antibodies now, and that includes small molecules that inhibit the virus. There's one, I think Merck has recently gotten emergency use authorization for that drug in a couple of countries. And that's basically just a small molecule medication that you can take. It also inhibits the virus, which means it also needs to be given early. And there seems to be another drug coming from Pfizer that works similarly. Again, all of these things have to be given early. in order to work because they target the virus. And in this late stage of the disease, targeting the virus has not been shown to be very effective. In the late stage, the immune system does the damage. But there is more and more of these options coming. What we really are hoping for is monoclonal antibodies that have a long half-life, that can be given prophylactically, and that then would provide protection for several months. And Regeneron, as I've shown you, has something like that in development, and we hope that that would become available widely soon. There's ways to, usually antibodies have a half-life of about 21 days, but there are ways now to actually extend that half-life to many months, and that would allow it to, give a transfusion of these antibodies every few months and basically passively protect people from infection and disease.
Okay, are you going to read these, Amber?
Yeah, I'll read them for you. So we have 2 questions in the chat. It says, what would you advise for people who got a non-US vaccine, AstraZeneca, Sinopharm? Do all need a new vaccine or only those at high-risk age, medical conditions?
All right. So currently what the Europeans do with AstraZeneca, at least a lot of European countries, they recommend a booster dose with an mRNA vaccine. AstraZeneca has been working well, and there is stable protection also against Delta, but the protection is lower as what you would see with Moderna or Pfizer. It's solid, but it's not as good. And so I think somebody who comes in from Europe and has 2 AstraZeneca vaccinations should probably consider getting at least 1 shot of an mRNA vaccine. For Sinovac, Sinopharm, and all these inactivated vaccines, I think the same is true. The vaccine effectiveness or vaccine efficacy of these vaccines is lower than what we see with AstraZeneca, for example. So it's even a little bit lower than that. Again, these vaccines do work. There's extensive studies from Brazil that they cut down on severe infections quite a bit. But it's also, I think, a good idea to get 1 or 2 shots of mRNA vaccine when you only had an inactivated vaccine.
There's one more question. It says, really excellent and informative presentation. Thank you so much for sharing all of the data and different perspectives on COVID vaccines and immunity. Can you speak more on your last point and which lab Sinai or our is now bioreference offer a quantitative spike, a nucleocapsid, or other COVID antibody testing? And have you envision these being used going forward with regard to correlated of protection of vaccinations in a primary care setting for healthcare workers, like we do for MMR, varicella, and hepatitis B?
Yeah, there's a number of quantitative tests out there. Sinai has one, LabCorp has one too, right? So they're available. One needs to be careful to use a test that measures antibodies to the spike and not to the nucleocapsid. Nucleocapsid is helpful if you want to establish if somebody was infected or not, but it doesn't help you measuring immunity or immune responses induced by the vaccine, just simply because there's no nucleoprotein in the vaccine. In terms of the usefulness and kind of how to do that. So we have a problem in the US, and that is that we haven't, most of these assays have not been converted to international units yet. And so the international units would then allow you to cross compare from, for example, what LabCorp measures, you get a value and oh, this is so and so many international units. And then you could compare that to the level of protection that studies got because they report out in international units, right? And so in the US, we're a little bit behind in converting to international units. And that's not necessarily the fault of labs that develop these assays or labs that run these assays. It's basically the FDA sitting on applications to change these tests, and they're just not moving because they're completely overwhelmed at the moment. Once that is done, I think it's getting easier. And yes, I see it in the way that, we titer antibody titers against hepatitis A, hepatitis B, other things, and then we vaccinate if the titer is too low. And I see that that is something that could be done and implemented for COVID-19 as well.
How can we know if current antibody testing, such as bioreference, have any value for our patients' decision making?
Well, again, you need a spike test. I don't know what the bioreference is, honestly. It needs to be a test that measures antibodies to the spike. Right now, I think the value is really for if you have patients who have certain types of cancers, who get biologics that are immunosuppressive, and so on and so forth. And in those cases, there's a lot of value in checking after vaccination, did this person make an immune response or not? And I think that's a very important point. If there is no antibody response after vaccination, I think it is certainly a good idea to probably offer a booster dose, or to have a discussion with the patient how to protect the patient from getting exposed to SARS-CoV-2, and also to make sure that you discuss with the patient that if there is an infection, that person needs antibody treatment right away, right? So for those kind of things, the antibody tests at the moment are super useful already. In terms of, at what titer should you get a booster, I think, as I said, I expect recommendations relatively soon for that, but we need to convert to international units across the system, and we need an official recommendation probably from the CDC.
Can you speak to the recommendation to mix and match? Is it more important to mix Janssen vaccine, but maintaining boosting with mRNA vaccine?
I think for booster dose with the mRNA vaccines, it doesn't matter much. If you got the Moderna and you get Moderna again, that's great. If you get Pfizer as a booster, that's fine. The other way around, if you get Pfizer and get Pfizer, that's good. If you get Pfizer and then you get Moderna, that's also fine. But I think if you get Johnson & Johnson, you need to get an mRNA vaccine as booster. I think that is very important. We see if you give a second dose of Johnson & Johnson, yeah, there is an induction of an immune response, but certainly not as strong as if you get an mRNA vaccine. I would basically say that get an mRNA vaccine, which one doesn't matter, but it should be an mRNA vaccine.
I'm going to jump in with a question, if you just, before we finish, we always like to look at some of the context in which all of this stuff takes place. So just maybe a quick two-part question. One is, what goes on with the approvals and lack of approvals and emergency use authorizations, and why some things get approved before other things? I mean, we've had Moderna now and Pfizer, and yet Pfizer's getting approved for all this stuff and Moderna isn't, and we get asked a lot of questions about that. And then the second part's going to ask you about the patent stuff that's going on now, which seems really crazy. Huge, billions of dollars going in from the government money into these corporations to allow them to develop these vaccines, and them claiming patent rights and making them less accessible to the public, and especially to the international public, which is, such a risk. I mean, if we don't eliminate COVID across the world, we're not going to eliminate it in the United States. It's just, so would you, I don't know if you consider this outside of your spectrum of things you're comfortable with, but I'd love to hear your opinion on both of those.
I think for the first question, I have some insights. For the second question, I have an opinion. The first one is it's usually up to these companies. I said the FDA is overwhelmed and they have trouble actually keeping up. But with those things that actually really matter, including full authorization of these vaccines now and authorization of vaccines for kids, of course, that's their main focus. That's what they do right now, and that's what they do quickly. In terms of the decisions of getting a full license and who's coming first, Moderna or Pfizer, this really has to do with who files first. Pfizer was always quicker than Moderna. Moderna usually talked more and had more press releases, and Pfizer filed quicker. And they filed for the full license before Moderna, and they got it, and Moderna is still waiting for it. And the same is true for vaccines for children, right? So Pfizer filed much earlier than Moderna. Moderna now has data, and they are, I don't know if they filed already or if they're planning to file. And so those are the differences, right? And the same is true when we look at Novavax. Novavax, they filed, they actually didn't file yet. They're waiting for, I think, some feedback from the FDA regarding their production process, at least in the US. AstraZeneca never filed in the US. They never filed an application for a license or an emergency use authorization. So in those cases, it's really the question, what did the company do and when did they do it? And then the FDA responds to it. So there is no preference for one or the other. In these cases, they move quickly if the applications are filed. The other question about the IP, that's a very good question. There's actually a number of things that need to be considered. The first one is the IP situation for the mRNA technology itself is not clear. So I would expect that there is going to be a lot of arguing and a lot of legal fights once the pandemic is over, because there's a number of players, University of Pennsylvania, Moderna, Pfizer, BioNTech in Europe, CureVac. There's a lot of players who have IP, and it's not settled at all. But then the other problem is there was this modification to the spike protein that came out of the NIH, of the Vaccine Research Center, this introduction of 2 prolines that stabilizes the spike. And a lot of vaccine companies are using that, including Moderna and Pfizer. And Pfizer licensed that technology from the NIH, and so did, for example, Novavax. Moderna never did; they just used it, right? Because they assumed when they work together, if they work together with the NIH, they have the rights to that. And then lately, we have seen that Moderna filed IP on vaccine constructs that were co-developed with the NIH, but they left out the inventors on the NIH side, and that has that's not just about now. That argument is not just about the NIH scientists saying, "Oh, we were left out. That's unfair." It is also a question about rights, because if the NIH is co-inventor, they have a lot of rights on these vaccines. If they're not co-inventor, they have no rights, and Moderna can do whatever they want. And that brings us to the bigger problem here, which is kind of sharing technology. And I think there is an IP component to that, but what is often overlooked, if you look at global vaccine production and global vaccine needs, it's not just IP, it's also technology transfer. So what you would want to have is companies like Moderna or Pfizer actually engaging in technology transfer with vaccine companies in low and middle income countries and help them to basically produce these vaccines there. And it's not just the IP, it's not just saying, oh, you're not allowed to do that, but it really needs a proactive movement from Pfizer, Moderna, and other companies to actually help companies in low and middle income countries to get the technology going, because if you don't know how to do that, you cannot establish that, right? So it's not just the IP, but it's also technology transfer that's needed. And you're absolutely right. We need to make sure that globally we have enough vaccine to basically vaccinate everybody, because if there are countries that have low vaccination rates and all of a sudden they have waves of infections again, there might be all kinds of variants that come out of that, and they might come back to the US. And even if we then are highly vaccinated, they might still cause issues, right? And so I think it's smart to make sure that everybody has access to the vaccine everywhere on this planet.
Dr. Kalman, you're muted.
Yeah, it's still shocking to me that so much of the research is publicly funded, but that the results of the research don't end up in the public domain where they can be shared. And I think, we're going to have to look at that at some point. I mean, even in the face of a global pandemic, like, we've never seen in our lifetime, we're still struggling with, ownership, private ownership of information and the production of the vaccine. So I know you can stay on for a little while, and so can some interested people. There's I see one more question. Did
Yes.
you want to read that, Amber?
Sure, this is the last question. It says, any plans for Sinai or New York City Health Department or Board of Education to study vaccine effectiveness in school? My 5-year-old daughter just got vaccinated yesterday, given at her school, similar to the Sinai studies done with healthcare workers last year.
So we are not planning to enroll school kids in our study. There's a reason why we did this with healthcare workers, because it's easy for them to come in, for study visits and so on and so forth, because they're at the main campus. But I'm not familiar with the details, but I'm 100% sure that New York State is planning to monitor that. Similar to what they did now, I showed you this one study where they look at vaccine effectiveness every week. They can stratify that by age, and they did before. And I'm pretty sure that they will include the age group 5 to 11 into these studies as well.
Great. Ray, did you have a question?
No, this is just an exceptional presentation that answers some questions and leaves some open. So thank you, Florian. This is really great.
Yeah, thank you so much. We'll have you back again in a few months when we really need some periodic updates, and we appreciate the fact that you're willing to come back so quickly and update us. I think this has really been superb. So appreciate it.
Thank you very much for the invitation. I hope I don't have to come back. It would be nice if all of this just goes away and nobody's interested in virology anymore. That's what I really hope for.
Yeah,
I mean, yeah, those comparisons that you showed with measles and mumps and stuff are astounding, right? In terms of infectivity, and but also the fact that those immunizations seem to be pretty much lifelong, right? And so there's so much we still have to learn to figure out how to, what the differences are in the way all of this stuff happens. I mean, you would expect something that's so much more infective to be harder to control, and yet it seems to be easier to control. So there's we'll be glad to hear chapter 3 at some point in the near future. So thank you very much again.
Thank you. Have a nice day.
Hey, everybody.
Take care, Florian.
Thank you.